which is specimen J. Specimen J is what? Egg, a section of boiled egg, a longitudinal section of boiled egg with shell. The shell will be intact. That is specimen J. Now, in this case, I always encourage students to give a call for experience, a call for what? Experience, because here you are expected to draw. It's possible that they will expect you to draw the egg. So in this case, why I said it's a call for experience. Now, when you boil the egg and cut it open, you find out that you will not see the chalaza. The chalaza will not be seen. But then what you are likely to see there is something that looks like, because I took some time to do this last night, and then I know I'm not going to leave it to this morning. So in biology drawing, you draw what you see and not what you know. You draw what you see and not what you know. Because I believe definitely you are likely to draw this thing. So my drawing is wonderful. This is the yolk. So now remember the shell is intact. Then this place is the shell. And then after the shell, you have what we call the shell membrane. And then the next one we have I have the yolk. This is the yolk. The other one is the albumin. Then we have this other one is the egg cell membrane. Then this side, this end of it is the air space. This is the airspace. Now, this airspace actually is where the air is trapped. During the development of this egg, they tend to take up oxygen from there. Now, the yolk is what supply was nutrient until maturity. Now, we have the shell, the egg shell, and the egg shell membrane, which is just immediately after as you're cracking it, you notice there's something around it. That little white substance is the shell membrane, and after that, we get the egg cell membrane. Then the other one that is white because it has been cooked, which is the protein part of it. As a result of what the nature of the protein, we have white substance inside here surrounding the yolk, which is the albumin. Albumin is there too. So this is what I think that you are going to be looking. Here we have the specimen for the 2025 WAEC biology practical exam, and the first in the list is a specimen A, which is bean swivel, and the last in the list is specimen H. So it's specimen G, which is the boiled egg, section of the boiled egg. That's have to boil the egg, and then you cut it with the shell intact. What it simply means, looking at that, it simply means that they are likely to be what? the kind of drawing of the internal structure of the egg. So then before I go ahead, anyway, let's just go ahead and do the thing. The first thing I want us to look at here is a specimen A. Specimen A, we have specimen A. Specimen A, which is the beans swivel. Bean swivel belong to the phylum. The phylum we call arthropods. Arthropods, that's the phylum, and the class is what? Insecta. And then, and it's also classified under the what? We call it Pterygota, which is winged insects. Winged insects. The other one that's not winged, we call it Aprigota. The winged one, unwinged insect. This one is for unwinged insects. And the bean weevil is known to be a very important pest of beans. It attacks beans. The adults eat the pollen grains, while the larvae feed on the pods and they seed. They drill hole into them, they make holes in them while they are feeding on them. They even live inside it until they get to maturity. Now the next specimen we have is specimen B, which is what? Beans with holes. Beans with holes. Beans with holes. This is as a result of what? The attack of specimen A. The specimen A, which is the bean weevil, normally make holes in beans, making it non-viable. The viability will be reduced, and then the market value will be reduced, and the same beans will not be very good for the market. That means the owner, perhaps the farmer might not be able to make good profit from it. So the next specimen we have is specimen C. Specimen C is likely to, I can say, one of the areas of interest in this specimen. Specimen C, which is specimen C is what? Waterleaf plants. This waterleaf plant will be in a hole, both the root and the shoot will be together. That's the whole plant. So these waterleaf plants will not need to be what? Dicotyledon. It's a dicotyledonal plant, and also now the leaf shape is what we call obivert. And the leaf margin is what we call the inter. That is what we call the leaf margin, because why we call it so is because it's very, very smooth. It does not have a saw-like edges. The margin is very smooth. We call it inter. So the next thing we're going to be looking at in this plant, we call it what am I saying, sir? The botanical name is Talecum triangulata. And then the next thing I want us to look at, because I believe that in this plant, the plant, having a whole plant is an opportunity for you to learn about the function of all the parts of the plant. In other words, we are going to be making a drawing of these plants so that we'll be able to look at them. So this is the plant. Here is the terminal bud. We have the flower, we have the leaf, the auxiliary bud. The terminal bud is the one that grows continuously. Now the flower is the reproductive part of the plant. The leaf is the area that manufactures the food through photosynthesis. Now we have auxiliary bud, the area that the bud that this auxiliary bud grows into what branches. Now we have the fruit. The fruit is already stored food. Now we have the node, which is the region where the leaf is attached. We have also internode, which is the distance between the node, one node to another. Now we have the stem, which is holding the whole plant, the short, the leaf, the branches in a position. Instead the leaf in a position they can photosynthesize. Now we go down here, we have the branch. This should be the branch roots. We have branch roots, which is for absorption of what? Water and mineral salt. Now we have the taproot system telling you that the plant is what? Diocotyledonous plant, unlike what we have in monocot, which is what? The fibrous root system. This plant has what? Taproot system. And then the vein, it has venated what? Leaves, meaning that they are not parallel. They are not parallel leaves, parallel veins, because as a result of what? They are what? Diocotyledonous plants. So this is what we want to look at here now. The next specimen we'll be looking at should be specimen D. The specimen D is another important area. But now there's something I'm going to let you know before leaving this very one, because it seems that in this exam you are going to be doing a lot of drawings. At least it's likely that you will make about 2 or 3 drawings in this exam. So based on this one, whenever you're making your drawing with the length given to you, make sure you draw effectively. Make sure you have the actual length. For example, since you are doing this, you measure out that length, do it effectively, and make sure you also get the magnification. The magnification is also very important. So the magnification is just the image of the length of this image you have drawn over what? The original image. Then you get your magnification. Anything you have there is your what? Magnification. Where you're making this drawing, make sure since they give you a distance, you can just take it. Let's say we are drawing this. Just make your drawing, make it there. It's not fine art drawing. The freehand sketching is enough to give you what you need to know. You need to draw. Just a freehand sketching, you get what you want. Put all the labelings where it's supposed to be, the fruit, the flowers, all the rest of them. Just do that. Let's say this is our 10 cm. Perhaps if you are doing such thing, this is our 10 cm. You just make sure you measure it out, and then make sure that whatever you're drawing is within that. Because most times they give you this based on 10 cm to 10 cm. When it's given like that, I always encourage students to use the biggest one, that the longest length, so that in a case where you fall short, you might not fall low. Because if you take the lowest length, you might also fall short, and then you lose out completely. Because if you're giving, let's say you're giving 8 cm to 10 cm, and you're drawing within the range of 8.5 or 9.5, you are still within the range. But when you choose the 8 cm because it's too small, if you fall within 7.9, you are out of the range, so you might be penalized. That means you are losing some marks. So that's why I always encourage them to take the last number. So we're going to be looking at the next specimen. The next specimen, which is specimen D. The specimen D is what we are going to be looking at now. Specimen D So this is the Quill feather specimen D. Now this middle part of it is what we call the ratchets. The ratchets. And then we have, or you call it the shaft. Now, just from the ratchets you have the barbs. The barbs that's coming out there. These barbs are also, they interlock each other with by what we call the what? There's an interlocking structure which we call the bubble. The bubble is what they use to interlock each other so that they will give a very good resistance to the wind when they are flying. So this one down here, all of them, the barbs, the rat is all those areas joined together will give us what we call the vein. The vein, and down there, immediately after the vein, we have what we call the superior umbilicus, and then we have also aftershave. We have the calamus and the inferior umbilicus. Here is hollow. That means there's hole in it, a kind of hole inside it. All of them give us what we call the quill. Now, what is this quill? It's adapted for what? Flight. It helps the bird to fly effectively. In this quill, in this feather, there's other feather in the bird. There are other feather in the bird. The one we call the corbels. The corbels. The corbel feathers. We have another one we call the down feather. The down feather, and then we have a phyllomy. Phyllomy is the one that remains after removing all the feathers in the bird. It's like a kind of a hair-like structure you see after removing the covert, the quill, and the down feather. Phyllomy is the one that remains that look hair-like structure you see in the bird after dressing it, when you're dressing it. So these are some other feathers we have in the bird. I want you to take note of this very well. So the next specimen we have here, is specimen D. The specimen D is the pineapple fruit. The pineapple fruit, that's what we have. Specimen D, which is specimen D, is it the specimen E, which is pineapple fruits. Pineapple fruits. Pineapple fruit is an example of fruit we call a composite. Composite fruits. This composite fruit is formed or developed as a result of many flowers coming together, very close together. They are very close together to itself to form this type of fruit. And these fruits, we have it's of two types. The first one is the one we call cirrhosis, and the next one is the one we call Sinochrome. Sinochrome is another one. The example of this cirrhosis is the pineapple, and this other one, the example of them is the fig. That's the example of what we have here. So these are something you should note about them, because you never can tell what will be coming out here. Just like I'm trying to guide you in some areas where they need be. But this pineapple in question will be cut longitudinally. When you cut it longitudinally, then that's what you're going to use for the exam. Now, that meaning that it's likely it might be drawn, they might require you to draw it so that you see the inside of it. I think we should do the drawing to give you a picture of what it's like. So this is the longitudinal section drawing of the pineapple. We have the crown leaf, we have the remains of the rats, petals, style, all of them are what we have around here. And inside here we have the swollen stalk of what inflorescence. Now we have this other region is combination of the ovary wall, receptacle, flower stalk, and all of them form what we call the edible part of it. So the next specimen we'll be going to look at here is the specimen F. Specimen F. We're going to be looking at specimen F. Specimen F, which is orange fruits. This orange fruit will be cut transversely. We're going to be cutting it. Now this is a sketch of what we expected to do. That is what we are looking at. That's likely to come out now, because you are going to see an orange that is cut transversely. So in this transverse section of the orange, you can see the picture of what you have. Now orange fruit is a type of fruit, is a type of berry fruit. It's a type of berry fruit. This berry fruit is the one we call hispidum. The hispidum is a type of berry where the orange belongs to, and one of the peculiar things about it is that the epicap and mesocap are fused together, while in other berry like tomato, the endocap and mesocap are fused together. So in this one, another thing about the hispidum is that they have a hair-like succulent substance in them that is juicy, where you get the whole juice of the orange. You can also see it inside them. And then the presentation of this plant, of this fruit, is what acid presentation. That is what the orange has. So I would want to encourage you to look at this as you're doing your studies. You look into this and then pay attention to all this drawing. Like I told you, whenever you're making your drawing, make sure you do your drawing in such a way as required. So the next one we'll be looking at now is a specimen, G, which is raw egg. And in this raw egg is obvious that we are likely to be testing for protein, because if you look at the other substances, other requirements, you notice that something like sodium hydroxide is there. We have sodium hydroxide, we have copper sulfate there. Now, for testing for protein, you need these two things. When you prepare your substance, when you prepare it, testing for protein, when you prepare all this, what you do is that adding sodium hydroxide to the sample, and then you add copper sulfate, copper 2 tetrasulfate 6. Then what you have is a purple color. That purple color is a positive test for what? Protein. That's the positive test for what? Protein. So specimen G is the next one we're looking at. In this specimen G, well, what you are going to be doing here is to test for what? Protein in egg. Because there we have a raw egg, and if you look at the other reagents given, you find out that you have sodium hydroxide and copper 2 tetrasulfate 6 there, meaning that, and the next after this is what? Water in a test tube. You understand? Water in a test tube. That's the specimen GH. The specimen H is what? Water in a test tube. So just looking at that, although you never can tell, but then me looking at that, I feel that there's possibility of testing for protein because of what I saw in the other reagents requirements. So based on that, that's what I'm going to be doing at this moment. You're going to watch. I have my sodium hydroxide, I have my copper sulfate solution, and also this is the sodium hydroxide in a test tube. So what we're going to do now is to get the this is already egg dissolved. What you do is that you prepare this, you get the white part of the egg, and then you add some water to it. So when you add some water to it, then it's ready for this experiment. So what we are doing now is that I'm going to take a portion of it. After this portion of it, too, the next thing I will do is that I will add sodium hydroxide to it. Adding sodium hydroxide to it, that's how we test for protein in egg. I've added sodium hydroxide in it. So this is the mixture. This is the mixture. The next thing I add copper sulfate. Copper sulfate, that's copper 2 tetrasulfate 6 into it. So you see what you're going to get. Can see the purple color. This purple color is a confirmation that we have what? That's a protein in this egg. This is the confirmation. This is how we confirm that what egg contains that protein in a substance. So this is one of the two. So that's what I think you might be expecting. They might be expecting you to do in that exam. So you can see the thing, the purple color has really come out very, very well. So let's continue the test. So the next one is the specimen. The next one we have, the next one is what? Water. That specimen H is just water in a test tube. In a test tube, water in a test tube. Water in a test tube. That's what we have our specimen H, water. In a test tube, water in a test tube. So you know water is very, very important in what? In digestion, in transportation of materials in plants and even in animals, because about 70% of what we have in our body, they are made up of water, and that water is playing a very important role in digestion. Even the blood we have also contain water. In fact, water happens to be a medium of what? Medium of transport, something like in the organism, in plants and animals. So the water is very, very important. Then we go down to the last specimen that we have here, which is specimen J. Specimen J is what? Egg. A section of boiled egg. A longitudinal section of boiled egg with shell. The shell will be intact. That is specimen J. Now, in this case, I always encourage students to use a call for experience. A call for what? Experience. Because here you are expected to draw. It's possible that they will expect you to draw the egg. So in this case, why I said is a call for experience. Now, when you boil the egg and cut it open, you find out that you will not see the chalaza. The chalaza will not be seen. But then what you are likely to see there is something that looks like, because I took some time to do this last night, and then I know I'm not going to leave it to this morning. So in biology drawing, you draw what you see and not what you know. You draw what you see and not what you know. Because I believe definitely you are likely to draw this thing. So my drawing is wonderful. This is the yolk. So now remember the shell is intact. Then this place is the shell. And then after the shell, you have what we call the shell membrane. And then the next one we have there, I have the yolk. This is the yolk. The other one is a albumin. Then we have this other one is the egg cell membrane. Then this side, this end of it is the airspace. Airspace. This is the airspace. Now, this airspace actually is where the air is trapped during the development of this egg. They tend to take up oxygen from there. Now, the yolk is what supplies was nutrient until maturity. Now, we have the shell, the egg shell, and the egg uh, shell membrane, which is just immediately after as you're cracking it, you notice the something around it. That little white substance is the shell membrane, and after that, we get the um, egg cell membrane. Then the other one that is white because it has been cooked, which is the protein part of it. As a result of what the nature of the protein, we have white substance inside here surrounding the yolk, which is the albumin. Albumin is there too. So this is what I think that you are going to be looking at, and then you pay attention. Thank you so much if you have come this far, and I want you to leave any question you want to ask us in the comment section. I will always attend to you. And then remember to share and subscribe if you're joining us for the first time. Thank you so much.